Good evening, and welcome to this year's virtual Biotech 101. My name is Neil Lamb, and I have the incredible pleasure of being your host for the next 4 weeks. Now, typically, we would actually all be together here on the campus of Hudson Alpha in the Jackson Center, enjoying cookies and coffee and fellowship. But the pandemic had other plans, so we're going to do this virtually. So I'm imagining you on the other side of that camera, maybe sitting on the sofa, maybe sitting in your favorite chair, maybe at the kitchen table. Maybe you've got a plate of cookies and a glass of milk. Maybe you've got some pedophores and a glass of wine or a cup of coffee. Whatever you've got, wherever you are, thank you for joining us. I'm really glad that you are here. We're going to have a fantastic time over these next 4 weeks. We're going to talk about the amazing world of biotech and genomics and DNA. Now, here's what we're going to cover. Tonight, we're going to focus on the basics. What is DNA? What happens when DNA changes? What are cells and chromosomes? I'm going to give you some general vocabulary to work from. Hopefully you've got the electronic version of the handouts. Every week we'll send the handouts in advance so you can follow through. You can follow through that PDF maybe on your tablet, or if you want, you can print those out. I would recommend you print them out in black and white because I don't want to use up all your colored ink. After this week, next week we're going to talk about ag and how changes in DNA impact the plants that we use for food and for feed and for fuel and for livestock. The third week, we're going to talk about cancer, which is something that touches almost everybody in the world, and about how cancer is a genetic disease and how changes in DNA that we acquire over time increase our risk of cancer. And then this wouldn't be 2021 if we didn't spend at least a little bit of time talking about the pandemic. So our fourth Tuesday night, we'll talk about COVID, what we've learned over the last 18 months and what we still don't know. You are watching this on YouTube Live, so you have the opportunity in the chat to type in questions or comments. My team is actually monitoring that, and we're going to stop periodically throughout the night and give you a chance. They'll send me those questions and I'll answer those questions live. So feel free to ask questions at any point in time. All right, so before we jump into the content, I want to thank Twickenham Advisors, who is this year's presenting sponsor for Biotech 101. I'm so grateful that they believe in the work that we do and that they provided the funding so that we can bring Biotech 101 in virtual format to you at no cost to you whatsoever. I also want to acknowledge our other sponsors, the Jackson Center and the Hudson Alpha Alumni Association, and just say thank you to everybody that supports the work that goes on here at Hudson Alpha, especially the work of the Education Outreach team. A little bit later on tonight, we'll talk about Hudson Alpha and what it is and does and how all the pieces fit together. That'll be the second part of our time together. But let's start with this. I'm on the campus of the Hudson Alpha Institute for Biotechnology. I'm in the library of the Education Outreach space. We've got some great props around us that we'll pull in as we work through tonight. But Hudson Alpha's official name is the Hudson Alpha Institute for Biotechnology. So what is biotechnology? Well, you could go to the dictionary or you could go to Google. I think that would probably be the more appropriate thing. Most of us probably no longer own dictionaries. But this is a definition for biotechnology, using biological systems, organisms, or processes to create products that improve our environment and quality of life. Now that's a mouthful. There's a lot of words there. So let me give you some examples around that. And let's start with this right here. This is common yeast. This is baker's yeast. You probably have this at home in a little packet that might be in your pantry or maybe in your refrigerator if you are a baker. If you aren't a baker, you probably depend on yeast for lots of things. For example, yeast are living organisms, and one of the things that yeast do is they take in sugars and they convert those sugars into carbon dioxide and into ethanol. And so we use yeast to turn flour and water and a little bit of sugar into delicious baked bread. And we use yeast to take grape juice and to turn it into wine. That's an example of ancient biotechnology, using something living, something biological, yeast in this case, to convert one product into another. Here's another example. You can take milk. That can be cow's milk, sheep's milk, goat's milk, camel milk, whatever kind of milk you want. And in a vat at a slightly warmer temperature, you can add an enzyme. And we'll talk about enzymes in just a minute. You can add an enzyme called rennet. And rennet is typically found in the stomach of a nursing calf. So we're not going to talk about how you actually have to get that. That would be an episode of Dirty Jobs, not an episode of Biotech 101. But you can add that to the milk, and it causes the milk to separate into curds and whey. And then you can pack the curds down, and you end up with cheese. So the process of cheese making is another example of ancient biotechnology. So is making sauerkraut and most methods of food preservation. But now let's take biotechnology and talk about it in terms of today. And typically that means that we need to start with this molecule right here, which is DNA, deoxyribonucleic acid, the famous twisted double helix. DNA is found in all living things. You and me, the food we eat, the plants, the trees, the frogs, cows, every living thing contains genetic information, contains this DNA molecule. It's not surprising that we call it the molecule of life. And when we talk about the sequence of DNA that's found in you and me, we sometimes refer to it as a book of life. So you're going to hear me use that analogy, the concept of DNA as an instruction book. Sometimes I'll talk about it as a recipe book. All living things are made up of cells. And here's a typical cell. And let me also start by saying this is all animated, false colored. If you were to look at cells under the microscope, they would not have these purples and pinks and all these beautiful colors, at least not unless you added stain to them. But this is a typical cell. Could be a human cell, could be a frog cell. We're going to focus on this purple sphere in the center. That's your nucleus. And those white strands inside the nucleus, that's your DNA. Half your DNA you inherit from your mom, and half your DNA you inherit from your dad. Your mom's DNA comes in the egg, your dad's DNA comes in the sperm. When they come together at conception, the two halves recreate the full amount of genetic information. So this strand of spaghetti-looking material, this is your DNA. These are your chromosomes. You and I have 46 chromosomes. Each chromosome is an individual piece of our DNA. It's the way that our DNA is packaged. And they come in pairs. So there's a specific time in our cells, right before our cells get ready to divide, when they have to copy our DNA so that each of our cells gets a full set of DNA. It's a little bit like having to copy the phone book. And the chromosomes condense down and they become visible, and you actually can stain them. You can add a dye to them and you can take a picture of them. It's one of the few times you can actually see the genetic material inside our cells in a really clear way. So this is called a karyotype. And then we used to actually take these pictures and cut them out with scissors and then align them up and look for all of the pieces. Today, computers do that work for us. So it pairs it up really nicely, which is on the other side of the screen. And you can see we've got 23 pairs of chromosomes. So there are 2 copies of chromosome 1, one that comes from mom, one that comes from dad. 2 copies of chromosome 14, one that comes from mom, one that comes from dad. So they come in pairs, except for that set way down there in the bottom corner, the X and Y. Those are your sex chromosomes. And they are not necessarily always identical. Females have 2 copies of the X chromosome. Males have 1 copy of the X and 1 copy of the Y. And here's an electron micrograph that's been false colored just to show you the difference in size between the blue X chromosome and the tiny little yellow chromosome. So some of you might be sitting at home thinking, I always knew there was something missing from those males. And you would be right. The Y chromosome has very little genetic information. It contains the master switch that turns on a set of genetic pathways that lead to maleness. And it has a few genes that are involved in sperm formation and fertility. But that's about it. It's pretty much a genetic wasteland. And some of you are not potentially that surprised about it. Some of you might be thinking, though, I knew that we could always travel light. We don't have to take all that extra baggage. We can get by with just a tiny little bit. So again, females have 2 X chromosomes, males have 1 X and 1 Y. All right, let's dig a little bit deeper beyond the specific pieces and let's talk about size. Your DNA is a tiny, tiny molecule. So small that at its width, it is 80 billionths of an inch wide. 80 billionths of an inch. So let's just do some scaling. Let's say that the entire world, that DNA, we're going to make it the width, not 80 billionths of an inch, but we're going to make it the width of a pencil. So if we expand DNA to that size and we scale the rest of the world up accordingly, what would we be thinking about? Well, a carbon atom, and atoms are some of the smallest components of molecules, would become the size of a rice grain. Well, that doesn't necessarily help me if I don't really understand how big carbon atoms are to begin with. So let's look at this one. A red blood cell becomes the size of a one-story house if DNA scales up to the width of a pencil. Okay, now we're talking about something that I can begin to get my head around. The thickness of a human hair becomes the length of a football stadium if DNA scales up to the width of a pencil. And the height of an average human, if we scale up the same way, is the distance from New York to Los Angeles. So that just gives you a sense of how incredibly small this DNA molecule is, and that it is this amazing molecule that directs life and leads to all this beautiful variation in you and I, and yet it is so incredibly small inside our cells. Okay, so here's our double helix. We've now zoomed back into our cell. This twisted ladder, I'm going to untwist it for a second just to make it easier to see. And again, this is all animation. If you were to be able to visualize DNA, and it's so small that you can't see it under a microscope at this level, but if you could, it would not be blue and red and green and yellow. We've colored that just to make it easier for us to see. Here's an actual model of what your DNA looks like. And you can see the twisted ladder. I've untwisted it in this image. Each one of these little spheres is an atom of carbon or hydrogen or oxygen that make up these components. But the image on the screen, we've kind of unwrapped it, and you can see the four colors. You can also see that what's on one side isn't exactly matched by what's on the other. Every time you've got a blue on one side, you've got a red, and every time you've got a green, you've got a yellow. And we know, if you know what's on one side of the DNA, then you automatically know what's on the other because the nucleotides, the inner rungs, pair in a very specific way. So let's get rid of one side of the ladder just to make it easier to see. And there are four components: adenine, thymine, cytosine, and guanine. And we abbreviate them as A's, T's, C's, and G's. And they're just like the letters in that book. They provide that set of instructions. The order of the letters on the DNA rung is like the order of the letters in a book. The order provides meaning. Now, we can scale back out a little bit, and we can talk about a gene. And a gene is a stretch of DNA. It's a set of letters that provides instructions. So for example, you have a gene that tells your body how to make the protein insulin. You have another gene that tells your body how to make the proteins hemoglobin that carry oxygen in your red blood cells. And another one for collagen. Collagen is one of those things that's in between our bones and in our nose, and movie stars injected in their lips. Your DNA is full of genes, about 20,000 of these recipes, all that provide instructions inside your nucleus, inside your cells. All right, there's a lot of terminology there, so let's break that down just a bit. The order of the rungs has meaning. The letters, the sequence of the letters, provides instruction. And let me give you an example. So here are four letters, M-E-A-T. Together, that spells meat. Meat has a meaning. You might have just had a great steak for dinner or a hamburger. Or you might be a vegetarian, and so maybe you had tofu instead. But the order of those letters has meaning. Let's change the order of the letters. Same four letters, but I've put them in a different order. Tame, team, and meta all have a completely different meaning. And then the word at the end is complete nonsense. It's gibberish. The same is true for your DNA. The order of the letters is what provides meaning. And specifically, the order that we're concerned about is the order in your genes. So a gene is a set of instructions. Some genes are 1,000 letters long. Some genes are millions of letters long. And they provide instructions to your cells for how to make proteins or how to make molecules that regulate how your cells work. So your DNA, sets of instructions within your DNA are called genes. And those genes tell your body how to make things. In this instance, we're going to talk about proteins. And proteins are made up of a set of building blocks called amino acids. And you have 20 plus amino acids inside your cells, and the order in which they get put together and assembled and folded is what gives your protein their specific function. It's what gives your bones their structure. It's what gives your eyes and skin and hair their color. It's what allows the muscle cells in your heart to contract in a rhythmic way. It's those proteins. And those proteins, the instructions for how to build those proteins come from your DNA. We have about 20,000 genes in the human genome, the collection of our DNA. And we used to think, once upon a time, that we must have had 100,000 genes, because we knew that things like fruit flies had, a few, maybe 15,000 genes. A banana has about 20,000 genes. So we thought we must be so much more complex than a banana. No, you and I are about as complex as a banana. We have about as many genes. The complexity for you and I is how those proteins are created and modified and the specific way that all those pieces interact together. That's where the magic happens. All right, I've just given you a lot of vocabulary. So let's pause, let's take a step back and put it into the whole picture. So here are your 46 chromosomes, paired by size. And those chromosomes contain DNA that's been tightly wound, it's been packed down. It's like taking a long piece of thread and winding it smaller and smaller, packing it down, and your chromosomes contain that wound DNA. A gene is a specific stretch of that DNA that provides instructions. And I'm going to show you two genes. The gene for HBB, which is on chromosome 11, and the gene for HBA2, which is on chromosome 16. If you were to look at one of these genes, it would just be a whole set of A's, T's, C's, and G's. So a whole stretch of those letters of genetic information. And that specific set of letters provides instructions that tells your cell how to assemble these proteins. So this is hemoglobin. Hemoglobin carries oxygen in your red blood cells, and it's a complex made up of two beta subunits and two alpha subunits. So here are the instructions for the beta in red, and then the instructions for the alpha make your protein that's in blue. So this is how the whole picture, all the vocabulary hangs together. So when you hear people talk about your genome, they're talking about the whole collection of DNA or the set of DNA inside your cell. Really, we've got two genomes, the copy we inherit from mom and the copy we inherit from dad. When they talk about your chromosomes, they're talking about the way your DNA is broken up into distinct pieces. And when they talk about genes, it's a stretch of that DNA that provides instructions. And then the final product generally is a protein. Sometimes it's another regulatory model. We won't worry about that too much. So let's pause, because I've just given you a lot of content. And let's see if you've got questions. So let me check my phone and see if anything has come in. All right, no questions so far. Feel free to send those in. I want to show you something that I think is really cool. Back in the 1990s and early 2000s, we sequenced through We created the first sequence of the human genome. So that's looking at all 3 billion of those A's, T's, C's, and G's in the human cell. 3 billion letters. It was the first time we'd actually read them. It was an audacious goal when we were able to sequence maybe a few dozen letters at a time on a really good day in the lab to say, we're going to figure out how to sequence 3 billion letters and provide all the instructions for that. Some of my colleagues here at Hudson Alpha were involved in the sequencing of the Human Genome Project under the leadership of our current president, Dr. Rick Myers. Before Rick was president at Hudson Alpha, he was the chair of the Department of Genetics at Stanford, and he led part of the Human Genome Project. And they sequenced a set of chromosomes for the human genome. And just as a fantastic visual, they actually put the sequence into books. So this is chromosome 16. This is the sequence of chromosome 16. And I know that you're not able to see this. This probably looks like just a bunch of gray. These are tiny, tiny letters. This is probably like 2-point font size. Nothing but A's, T's, C's, and G's, page after page after page. There are 90 million letters in this book, in chromosome 16. 90 million bits of genetic information. This is 3% of the entire human genome. So just to give you a sense of the amount of genetic information in our cells, 90 million letters, 3% of the human genome. I'm going to pause for a water break. These lights are hot. All right, so let's shift now a second and let's connect the concept of DNA to the visible traits and diseases that we see. So traits, the characteristics that you and I can see, like eye color, are influenced by multiple genes. So there are multiple genes. You can see each one of them. Well, actually, you can see a handful of them here. These are the primary genes that are involved in the production of melanin, the pigment that appears in your eyes and in your skin and in your hair. So if you go back to your high school biology class and you talked about big B, and big B was brown and little b was blue, and people that were little b had blue eyes, and people that were big B, or big B, little b had brown eyes, that's not the way it works. Eye color is much more complicated than a single letter combination. There are multiple genes that all work together to influence the type and the amount of pigment that shows up in our eyes. It's not a simple process. On the flip side, here are the genes and their specific locations that are known to be associated with amyotrophic lateral sclerosis, Lou Gehrig's disease, a neuromuscular degenerative disease. There are multiple different genes that are involved, and at any one of them, changes in those genetic recipes can increase your risk of developing ALS. Many traits and many diseases are not simple one gene, one disease. Some are, but many aren't. And just because we've been talking about humans all this time, I want to remind us that all living things contain DNA. So these are the chromosomes. These are the 10 pairs of chromosomes from corn. And I'm showing you 3 different genes that are involved in converting the starch in corn into sugar. So if you've ever eaten field corn, which is what is often given to animals, you know field corn tastes nothing like that beautiful yellow, juicy, sweet corn that you get in July and August here in Alabama and in lots of other places as well. And field corn tastes the way it does because each of those kernels is full of starch, not sugar. But a specific change in any one of these genes increases the amount of starch that gets converted to sugar. So traits in plants, traits in livestock are controlled by genes just like they are when we talk about humans. Different cells activate different genes. So you've got 20,000 plus genes in your body. All 20,000 of those genes are not active in every single one of your cells. There's a handful of them that are active in all cells. They're called housekeeping genes. They provide the things that keep your cells, that allow your cells to interact with the environment, that regulate the amount of energy that they're taking in and the way that they're getting rid of waste and when it's time for them to grow and when it's time for them to divide. But most of your genes are not housekeeping genes, and they're only active in a subset of cells or at a specific time in development. Because again, they provide instructions for making proteins that your cell needs, maybe to be a muscle cell, or maybe to be a liver cell, or a skin cell. And so only a subset of those genes, and I've indicated those like tabs in our book of life, only a subset of those specific genes are active at any one time in any one cell type. And over the life of a cell, there may be genes that are turned on when a cell is newly created, newly divided, that are then turned off over time. As you and I develop from a conception, from a fertilized egg to a fetus, to a child, to an adult, there are sets of genes that are turned on and off at different points in our development. So DNA is the parts list. This is a carburetor, at least that's what I think it is. I am not mechanically minded. I actually had to ask my dad. He said, yes, this is a carburetor. But DNA is the equivalent of the parts list. It tells us, all the genes tell us each specific piece of the instructions. But they can't tell us by itself how the parts fit together. What happens when a part wears out? How do you fix a part? For that, you've got to gather more information. But the genome, sequencing the DNA, getting that set of genes, gives you the parts list. Let's talk a little bit about the genome inside of you and I. So this would be our human genome. In humans, only about 1 to 2% of all of our DNA are actually genetic instructions. That just seems so strange, doesn't it? That the vast, vast majority of those 3 billion letters are not instructions. They are not genes. Only 1 to 2% are. Much of the remainder is thought to be in some way, shape, fashion, or form regulatory. So it controls when those genes get turned on, when those genes get turned off. But a chunk of our DNA is probably evolutionary filler, left over from ancestral viruses that were in our cells, from other elements of DNA that are now completely non-functional. So 1 to 2% are the genes. Probably something like 7 to 15% more are the regulatory pieces. And then we've got all the rest of this that we aren't really 100% certain about what it is and what it does. The genomes of any two humans differ by less than 1%. Let me say that again. The genomes of any two people, you and I, our DNA differs by less than 1%. Doesn't matter where on the earth we come from. We are so much more alike at our molecular level than we are different. And those things that we tend to characterize each other on, our appearance, our eye color, hair color, skin color, our height, our body shape, that is a tiny, tiny fraction of the overall genetic instructions within us. I don't think you can see it, but I have a jar here that's made up of all red beads. There's 1,000 red beads, and there is 1 yellow bead. It's probably hard to see, but this gives you a sense of that 99.9 or 99.5% similarity, all the red beads, as opposed to that 1 yellow bead that talks about our genetic differences. Now, don't get me wrong, that's still 4 million genetic differences between you and I, but that's 4 million out of 3 billion overall bits of letters. Some of these letters, some of these differences are a single base, one of the rungs of the ladder. Some of these differences are thousands of letters long. And you can't always say that the length of the change correlates with the severity of the impact. Sometimes thousands of letters being missing has no impact at all. Sometimes a single letter change has a serious impact, like sickle cell anemia. One letter change in both copies of one of the hemoglobin genes. So we're talking a little bit about change. What happens when that DNA recipe changes? So what's the impact of that? So for us to look at that, I want to transition us to a different set of analogies. So we've been talking about an instruction book. I'm now going to shift us to another kind of instruction book, a recipe book. So I want you to think about a cookbook. I love to cook, and this is one of my favorite cookbooks because it talks all about the science behind cooking. Cooking is an art, but there is a significant amount of science to it. And the cookbook contains lots of individual recipes that tell you how to make a specific product. In this case, the recipe for French bread tells you how to make French bread. I want you to think about this storyline as for DNA, the cookbook is our genome. It's our full collection of genetic information. Genes are the recipes, and sometimes the cookbook includes a lot of other things that aren't genetic, like the front copyright information and the table of contents and dedications and how to fold napkins and how to choose what wine to serve. But it includes recipes, and the genes are the recipes. And then our final product, often proteins, but sometimes not, that's the final outcome. That's the equivalent of the French bread. So let's think about this in terms of the concept of a recipe. So this happens to be my grandmother Lamb's recipe for chocolate cake. At least I think it's her recipe for chocolate cake because Grandma sometimes left things out of her print recipes so that nobody could actually ever fully replicate her recipe. So this is the recipe I've got. We'll see if it's the actual exactly 100% identical. But you can see it uses sugar and flour and cocoa powder, all these things, chocolate frosting, which is a separate recipe, and that gives us a delicious chocolate cake. Moist, yummy, rich, delicious. Let's change the recipe a little bit and see what happens. So let's change brand A flour to brand B. This is the difference between like White Lily and Swan's Down flour. Now, my grandmother would say that that makes a big difference. She was very specific on White Lily flour. But at the end of the day, this probably looks and tastes just like a chocolate cake. That change really doesn't make a big difference. How about another change? Let's go with strawberry frosting. Okay. Is this still a cake? I think most of us would say yes, it still meets all the definition of a cake. I might like it better because I prefer strawberry. I might like it less because I prefer chocolate. It's a variation, but it still meets the definition of a cake. All right, let's change the recipe some more. Let's leave flour out entirely. Now at this point, I've got something that is a flat, probably pretty soupy, goopy mess, which most of us would agree no longer meets the definition of a cake. I've changed this recipe in a way that has created something that no longer resembles a cake. Now, I could also make other changes in this recipe. I could add a little too much sugar. I could add a little too much salt. I could add not put enough milk in, and I would change that as well by some of these smaller combinations of changes. But let's stick with this concept. So now let's go from our cooking recipe back to our book of life. And let's talk about what happens when the DNA recipe changes. So what happens when I start fiddling around with those genetic sets of instructions? You're going to see the same broad categories that we just talked about with our cake. So in many cases, you're going to see a completely silent change. There's no difference at all. This is like White Lily versus Swan's Down cake flour. The vast majority of the changes in your DNA fall into this category. They're completely silent. They make no difference whatsoever. You would never even know that they're there. Then you've got another category. This is called polymorphisms, and this is many forms. This is part of normal variation. This is what you see when you look, for example, at eye color, which we've already talked a little bit about. All the different patterns of pigment in the eyes. This is like chocolate versus strawberry cake. One is not better than the other. It's different. It still meets the functional definition of the pigment in our eye or in our hair or in our skin. And then we have changes that are called mutations. This is like the equivalent of I've left out the flour or I quadrupled the amount of milk. These are harmful. Now, if there are any biology teachers that are watching in the audience, I recognize that the official definition from a biology class, the definition of mutation is any DNA change, regardless of the outcome. In a clinical world, mutation takes on a slightly different term. It's a change that has a detrimental outcome. So we're going to talk about mutations as specific things that increase or decrease risk of disease. We're going to talk about polymorphisms and variants, when we talk about genetic changes that are not mutations, that are not harmful. The challenge here is that sometimes the definition of polymorphism and mutation is in the eye of the beholder. So as an example, the deaf community, many forms of deafness are caused by specific mutations in known and understood genes. But the deaf community would say, we don't have a disease. We sit on the spectrum of the ability to hear or not, but we have a vibrant language that allows us to communicate. We have a culture. We would not see ourselves as having a disease and needing a therapy because we have genetic mutations. Other people, even within the deaf community or outside the deaf community, might look at that in a very different way. The same is true when you talk about genetic mutations that may dramatically increase or decrease height. Individuals that have achondroplasia, which is a form of short-limbed dwarfism, have a specific genetic mutation that causes the long bones in their arms and legs not to develop fully. They would not necessarily say that they have a mutation and that they have a disease. So some of this really straddles between sets of definitions and the way that we interpret health and the way that we interpret disease. So we want to be careful before we throw around terms like mutant and mutation and think about the overall context with that. Let's talk about how we get this genetic variation, these changes. Most often, this happens when you have to replicate your DNA. So every time your cells divide, they have to make a complete copy of all their DNA so that the two daughter cells have full sets. So imagine those 90 million bases, which is only 3%, your cells have to faithfully make that copy every time your cells divide. And they're good, and they've got proofreading systems that help catch mistakes, but they aren't perfect. So every time your cells divide, there's a handful of new changes that appear that weren't present in the original copy. You can also get DNA change from DNA damage that doesn't get repaired. So for example, you think about ultraviolet light, the sun's rays, it actually damages the DNA, it causes breakage, it changes the letters, and your body has to identify that damage and then has to repair it back. And often it does a really good job, but not always. So sometimes mutation or DNA variation is the result of unrepaired or poorly repaired DNA damage. If this occurs in an egg or a sperm, in the cells that give rise to egg or sperm, and that egg or sperm is involved in a fertilization event, then that genetic variation has now been passed on to the next generation. And it's present in all of that individual's cells, including their own egg or sperm, and they'll pass it on, or they may pass it on to the next generation. If that change doesn't occur in an egg or a sperm cell, it occurs in a body cell, then that specific cell and all the cells that come from it will have the change, but the rest of the body won't. So let's say that it's a change that happens in a skin cell, but it doesn't happen in an egg or a sperm cell. It's not passed on. So this is the difference between a mutation that gets inherited from your parents and a mutation that you've acquired. And that'll become really important when we get to week 3 and we talk about cancer. Because cancer, yes, there are mutations you can inherit from mom or dad, but it's primarily a set of mutations that you acquire in a subset of your cells over your lifetime. All right, let's talk about the impact of those mutations. This might make you want to hit pause and run to the freezer to grab some ice cream or a frozen dairy treat, or maybe you want to run down the street to the nearest ice cream store. But for some of you, the thought of ice cream just makes your stomach hurt, literally, because you might be lactose intolerant, and dairy products give you incredible abdominal pain and great unhappiness. There's a genetic mutation that's associated with this, but interestingly, the people that as adults can digest milk and ice cream and dairy products, they carry the genetic change. For most of life on Earth, the genes that created the proteins that allowed infants to digest the sugars in milk, those genes shut off around the time that the baby or the infant or the young cow or whatever was weaned. So they were active when they were nursing and they were silenced afterwards. But in different populations of humans around the world at different time points, a mutation occurred, a DNA change occurred that caused those genes not to be shut off, but to stay active. So that even as adults, they could extract nutrients from milk. And that gave them an advantage because it's another source of energy for a food source, especially in a place where food might have been scarce. So those of you that are lactose intolerant, you are the ancestral version. The people that are lactose tolerant, you carry the genetic change. I already talked a little bit about pigmentation and skin color. There are about a dozen genes that provide this incredible spectrum of pigmentation. A dozen. That's it. That's a tiny, tiny fraction of the overall amount of genetic information that we bring to the table. And yet it is one of the things that we most often make decisions about based on that visible trait. 12 genes. 12 genes. And then we can talk about human height. There are hundreds, maybe even 1,000 genes that contribute to human height, each giving us tiny millimeters of height or subtracting height away. Yes, there are a handful of genetic changes that cause big differences in height. But in general, the thought that you could use genetic information to select for a 6.5 or a 7-foot tall basketball player is ludicrous because there are so many genes that are a part of that overall trait. We can also talk about the level of change, the level of mutation. Individuals that have Down syndrome, for example, instead of having 2 copies of their 21st chromosome, one that they get from mom and one that they get from dad, they have 3 copies of that chromosome. That's an extra copy. It's an extra 1.5 million letters, an extra few hundred recipes. But that extra throws off the process of development and leads to the characteristic set of symptoms that are associated with individuals with Down syndrome. Individuals with other chromosome anomalies have entire chromosomes, extra chromosomes, or missing chromosomes, or pieces of the chromosome that are extra or missing. So in that case, talking about genetic variation, you're talking about huge amounts of genetic data. In contrast, sickle cell anemia, which we've already mentioned, is 1 letter change in one of the hemoglobin genes. And that 1 letter change is enough to change the protein so that those red blood cells, when they aren't carrying oxygen, when they've given up their oxygen to healthy tissues, though that protein actually can stick together. It can form long strands of protein that distort the shape. So the red blood cell goes from pillowy donut shaped to sickle shaped. And then it gets clogged in arteries and it causes all sorts of pain and all sorts of other challenges. Most commonly found in populations of African descent, but not exclusively. So you find individuals from all ethnicities that can have the mutations that give rise to sickle cell. 1 letter. And then something complex like the development of the heart or the development of the brain is hundreds of genes all working together at different points in time, each contributing their portion of the recipe to make the overall tissue, to make the overall organ. And sometimes throwing that balance off just by a single change is enough to cause anomalies in development. Other times, it's more resilient than that, and a single change doesn't have any impact whatsoever. These machines are the workhorses of our genome. If I weren't standing on a wooden platform, I would bow before them because they are the heroes. They are the superheroes of the field of genomics. These are DNA sequencers, and they read that set of genetic recipes. So I can take a sample of blood, and I can collect the DNA from the white blood cells, and I can break it into pieces, and I can do some chemistry and put it in these machines, and they will give me the readout. They will read the sequence back out. It's a little bit like taking your favorite book and putting it in a wood chipper so it gets shredded into pieces, and then taking each little piece and transcribing, writing it out, and then figuring out how all the pieces get put together. They are amazing. These machines read about 150 letters at a time. This is called short read sequencing. These machines read thousands of letters at a time, maybe 12 or 15 or 18,000 letters at a time. It's called long read sequencing. In the year 2000, some of you were alive in 2000. You remember we were all, right before 2000, we were all getting ready for Y2K, and we had filled our bathtubs with water, and we had bought all this food because we thought that the world was going to shut down at midnight when the clock changed. If you weren't paying attention to Y2K and you were sequencing a human genome, it would have cost about $500 million to sequence the human genome. That is not pocket change. Today, because of these kinds of technologies, you're now talking about less than $1,000 to sequence a human genome. And you can do that in a matter of days. Now, that doesn't interpret your genome. That just gives you a whole set of A's, T's, C's, and G's. You still have to go through all the process of reading and interpreting it and making that work. And so at places like Hudson Alpha, places with sequencing machines around the world, this is what they do. It may be human DNA, it may be DNA from a plant, it might be DNA from a fish or from some other important organism, and they read the genome, they read that sequence. And once you've built the first set of sequences, you can then read somebody else's sequence and you can compare it to the reference. And you look for those changes. You find those 4 million changes in you and me. So this now really becomes an exercise in computation. You need huge amounts of computer storage space to store all the information and to process the information and to analyze the information and to look for those changes. So the field of genomics has a huge component that is informatics, that is computational. If you know young people that are interested in computers but also love science, this is an amazing field for them to go into, the concept of bioinformatics or computational biology. Because now we have more data than we know what to do with, and we have to parse through the data and dig through and look for the patterns in the data that tells us how to separate out the wheat, the important stuff, from the chaff, from the rest of the pieces. Which means we go from the parts list to the full instruction manual that tells us how all these pieces fit together, how the pieces come into, how they work together, what happens when part of them break or when they wear out, how you think about preventing that, the preventative maintenance around that. And that leads us to a concept called precision medicine. Some people call it individualized medicine, some people call it personalized medicine, but this is the idea, and this is a part of where medicine is going. If you can combine, if you can know your genetic risks, if you can identify, read through your DNA and identify the changes that are important because they increase or decrease your risk of disease, and you can combine that with your environmental risk factors, your lifestyle, the things that you've been exposed to, then you can determine overall risk susceptibility. So this is more than just sequencing your DNA and saying, oh, you've got these changes, but it's determining if you're at risk for developing cancer or neurodegenerative disease, or the field of pharmacogenomics, which is where I think most of us will interact with genomics first. And that's determining, are there certain medicines that don't work for me? Are there certain medicines that I need to change the dosage of? Because the way that your body uses medication, enzymes convert the drug that you've taken, the medication, into an active form so that your body, it can do what it needs to do in your body, and then other enzymes break it down. And changes in the DNA determine if those enzymes can do their job or not. So if you have a genetic change that means that you can never make a drug its active form, that drug does you no good. On the flip side, if you have a genetic change that means that you can convert that drug to an active form, but you break it down in your body after it's been active really slowly, and you're still taking the dosage twice a day every day, you may run the risk of having toxic levels of the medication in your body. So pharmacogenomics is, I think, one of the key places that we'll see the impact of genomics on human health. And that brings me to a really important point. DNA is not the sole determinant of who we are. So here's a doctor that says, take 2 genes and call me in the morning. That is not the way this works. And here's a guy who's just been arrested by the police who's mugged a guy in the alley and says, my genome made me do it. Our health, our personality, our behavior, our traits are a combination of our genetic and our environmental factors. There is a strong component for many diseases. Now, some diseases are very environmental, and some diseases are very, very genetic. But almost all of them have some combination of the two, maybe more of one than the other. And we have to be careful that we don't simply say, oh, I'm fated because I've got this genetic change that increases my risk of cancer. I'm going to get cancer, end of the story. Generally, that is not the way this works. On the flip side, you don't say, oh, I don't have the mutation that puts me at a higher risk of Alzheimer's, so I am completely in the clear. Again, that's not the way this works. Now, you don't have this in your slides because this paper actually was just published yesterday in the journal Nature. But it's a really fantastic commentary on the concept of disease risk and precision personalized medicine. And what's really nice, number one, it's not behind a paywall, so everybody can access it. And number two, it's really readable. It doesn't have a ton of jargon, and it is full of examples. So if you want a sense of where is this field going and how do you combine all this genetic information and your environmental information, which is what they show over here, your genetic variants and your environmental factors, to determine prediction about disease risk, and then measure over time what's happening in your body. This is not a one and done thing. I've gotten my risk factors, now I know what's going to happen, but it requires continuous monitoring and tweaking and adjusting. If you're interested in this field, this is a great paper that I would recommend that you take a look at. So screenshot this really quick so that you can find it, or you can also just go to the journal Nature. I think it's nature.com, and you can just type in personalized profiles for disease, and this should pop up. It was published yesterday. All right. Again, a whole lot of information that we've covered. Let's pause and let's see if we've got any questions. So John asks, is there a logical explanation for how genes occur on chromosomes? Are they organized in a specific manner? John, that is a really great question. Is there a specific way that our genes are organized so that all of the genes that are involved in brain development are on one chromosome and all the genes that are involved in liver development are on another chromosome? And that is not the way that it works. In some senses, it looks like it's a mishmash. You've got genes in multiple different places. Part of that is an evolutionary background that when you go all the way back to early forms of life that had many fewer chromosomes, the genes were arranged maybe in a specific order, but over time, those chromosomes got duplicated, or those genes actually moved, or some of those genes copied themselves. So for example, the genes that are involved in our ability to smell aroma, they are tiny genes. They're like 1,000 letters long. And sometimes you'll see a whole bunch of them right in a row, and they are copies of an original version that they themselves have undergone change over time. And those changes over time allow us to detect different aroma molecules in our nose. So sometimes they're grouped together from that historical duplication point. But you might also find another set of genes that are involved in olfaction, our ability to smell, on a completely different chromosome. So there is a rhyme or reason to it based on the evolutionary history way back in the tree of life. But at first glance, it sometimes looks like a complete jumble. But that's a great question. Thank you for asking that. And kudos, John, because you're the first person that asked a question. So well done. Gold star for you. All right. Let me shift gears a second. We've talked a lot about changes in DNA, how DNA impacts traits and disease. Let's now talk about the work that goes on here on the campus of Hudson Alpha and how that concept of biotechnology all fits together. I've been at Hudson Alpha almost since the beginning, not quite. I got here, some of you have heard me say this, I got here when the building was literally a hole in the ground. And the vision of Hudson Alpha is what captivated me and what continues to captivate me, the way that we think about the field and the way that we can contribute to the field. And that is due in very large part to these two gentlemen on the screen behind me. This is Jim Hudson and the late Lonnie McMillan. And Jim and Lonnie were the co-founders of Hudson Alpha. And they had a vision of a place where you could do world-class science, and in the same space, you could bring companies together that are building products based on the outcomes of that science. And that you could pull an education component together that is using the stories of the science and the work of the companies to engage the public and to train the next generation that works in both of those places. And that those would all mix and mesh within the same space. And there'd be a synergy and a sharing of ideas. And at the time, that was an incredibly rare idea. They were incredibly visionary for thinking about the space. And they brought the energy and the funding and the other support to the table to create Hudson Alpha. And our mission is to use biotechnology, there's that word that we've just defined and described, to improve life, to stimulate economic development, and to inspire the youth of Alabama to seek careers in science. So you can think of it like three partially overlapping circles. The research in the field of genomics, looking at DNA, trying to understand how changes in DNA impact human health, impact sustainable ag, sources of energy. Building an environment where companies want to come here, and they want to grow here, and they want to stay here, so that you are surrounded by this economic engine built around life science. And then training the future and creating a more genomically literate public. These are the research faculty at Hudson Alpha. And you can see underneath, these are all of the different things that they work on. So these are research scientists that were recruited from universities from around the country. Nick Cochran in the upper right-hand corner is our newest faculty member. He became a full-fledged faculty member earlier this month, and it's really exciting to have him on board. He works with neurodegenerative disease and brings some great strength. But each of these faculty run labs, and those labs have technicians and senior scientists and undergrads and graduate students, and they are seeking to understand the mystery of the genome, how changes in DNA lead to these different traits, and then how we can use that to better understand health and disease and how we can feed the world and how we can provide fuel. The Institute's a nonprofit research space. We are not on our own a company. There are companies on campus, but we're a nonprofit. And so we depend on funds that sometimes come from the research grants we write, funds that come from contract work that we do with other organizations, funds that come from places like the state of Alabama and legislative support that allow us to help put buildings on our campus or help grow our education program, and then finally from individual donors and corporate donations. That's how our work is done. There are 46 biotech companies on the campus. 46. Some of them are just in startup mode. Some of them have more than 100 employees. Some of them develop diagnostic tests. Some of them are developing products that are being used by labs and companies. Some of them are working in the field of agriculture. Some of them are working in the field of cancer. Some are developing new drugs. But each of them brings something to the table. Some of them are working on science that's been developed here at Hudson Alpha. Some of them are working on science that's been developed in other places. But there's a reason that they want to be here. There's a synergy. There's an interaction of being in this place where they actually can support each other, where they can grow their own, where they can identify potential funding to continue to grow. There are 4 main buildings on the Hudson Alpha campus. I'm coming to you right about here from this building. The main building right here, 601. The Jackson Center, where we would typically be for Biotech 101. And then 701, which contains space for multiple different companies. It contains some of our agricultural growth chambers. It contains our Smith Family Clinic for genomic medicine. Here's the bird's eye view of the entire campus. So again, you can see those same 4 buildings, and then you can see this beautiful McMillan Park. There are over 700 trees on McMillan Park, and it has a walking and a running trail that form a kilometer-long DNA double helix, which is kind of hard to see now that the trees have grown, but you can see it really nicely from this overhead view. There's multiple space on our 153-acre campus for growth. And in fact, right now, we are in the process of building the next 2 buildings on our campus. So a head house and greenhouse space for our growing ag program, and then the world headquarters for Discovery Life Science, one of the companies that's actually been with us since the early days and grown and merged with other groups. And they're going to now bring their world headquarters here to Hudson Alpha. It's exciting to see these buildings now come out of the ground and to, again, see the vision of Jim and Lonnie continue to come to fruition. The education outreach piece is what I have the pleasure of leading, and I have an amazing team of master educators and genetic counselors and support staff and individuals that all work together to create a more genomically literate society and to train the next generation of people that are going to work in the field of life science. We create kits that go in classrooms around the country and around the globe. We provide training to support educators to share this information with their students. We partner in clinical settings to explore how we bring genomics into a patient care setting, both from a research and from a clinic setting. We create immersive student experiences that let students put on a lab coat, put on the career, and see what it's like to work in this field. And then we build workforce programs that provide training and a set of skills to help students move to the next set of whatever the next step for them, whether that's moving on to an undergrad program or preparing to go into the workforce or heading on to graduate or other professional school. We do this in education for a number of reasons, but I want to tell you about one that's near and dear to me. So this is 7th grade Neil. I grew up in Chattanooga. I'm really jealous of all the hair that I had at that point in time. This is my 7th grade life science teacher. Her name was Frances Bowman. And Miss Bowman had this really cool piece of equipment in her 7th grade classroom, which was a microscope with 2 sets of eyepieces. So she could look in the microscope and a student could look in the microscope. Every time I looked in by myself, I was always seeing like eyelashes in the inside of my eyelid. I was never seeing what I was supposed to see. But that piece of equipment made sure I was looking at the right thing. And Miss Bowman said one day, I want everybody to bring in pond water or lake water or river water or a stream. I want you to bring in a sample of water from someplace other than your faucet. And so I brought in water from the pond up the street and we poured it in a little dish and put it under the microscope. And this is what I saw. This baggie of water that I thought was completely sterile, that looked like it had nothing in it, was teeming with life. And I was hooked in the field of science, in part because of this. Ms. Bowman had the right activity to engage me and the right piece of equipment. We try to do the same thing here in education at Hudson Alpha by creating activities that engage students in real-world ways that make them go, oh, and putting the equipment into a teacher's hands, or bringing students here where they can actually see that equipment firsthand if that's not something you can easily put into a classroom. That's part of the vision of what we do in education outreach. We truly believe that education is not knowledge that I am pouring into a student, but it is a fire of interest that my job is to ignite. Our job as educators is to be fire starters, and I mean that in the best possible sense of the word. So let me come back to our 3 mission areas here at the Institute. Each one of these mission areas has a specific area of focus. For example, our economic development team is dedicated to how do we recruit new companies to come to the Institute? And when they're here, how do we make sure that they have what they need to continue to move to the next level? How do we help grow these companies? How do we ensure that Hudson Alpha is a hub, a bioscience hub here in the Southeast? You've heard about some of our genomic research pieces and our education pieces. So each of us have mission-specific programs. But then there are programs that sit at the center of all three of these areas. And for us at the Institute, there are two key ones. I'm going to tell you just a little bit about it, and then we'll wrap up. One is in genomic health, and one is in sustainable ag. In genomic health, we have a huge amount of expertise on our research side in how we analyze DNA, and we look for changes in DNA that may be correlated with developmental delay or disorders, or neurodevelopmental issues, or neurodegenerative disease. Our researchers have a huge wealth of expertise in that. We have a clinic on campus, the Smith Family Clinic for Genomic Medicine, where we see patients and we can talk with them about, based on their symptoms, does it make sense to look at genetic information? Does it make sense to look at a subset of genes or a panel of genes, or to sequence the entire genome to try to identify what might be causing or related to their symptoms? And then we have an education group that works with how do we provide patient education. Our genetic counselors are the go-between between all this content and the individual patients themselves. And then how do we think about the way that we turn this into an opportunity, something that we can offer to other organizations? Our clinical service lab does this kind of testing, offers these sorts of tests, the sequencing of the genome, looking at cancer panels. And then our Health Alliance is an organization that falls under the broad umbrella of genomic health. How do we use all this information in ways that people actually find valuable? How do we give them information about their risk factors for certain diseases, their risk factors for adult onset cancer, or pharmacogenomics? How do all these components all fit together? And it pulls on all three of the mission areas at the Institute. The same sort of thing is true when we talk about ag. We live in a world that is increasing in population, yet is decreasing in the amount of farmable land. So how do we provide more food for people with less land in the middle of changing climate? The secret, the answers, are contained in the DNA of plants and crops. Plants grow everywhere in the world under the craziest, most extreme conditions. And the answer is because of the specific genetic changes that they have. So if we can identify those, often if it's present in one plant, it may be in another plant, or there are tools that you can use to make those changes. So how do we think about the concept of sustainable ag for the world around us? So here's our wrap up. Here are the key things that we've covered. DNA is the recipe of life. It's a set of instructions that provides meaning. It's stored in the nucleus of our cells as tightly wound structures called chromosomes. You and I have 46, 23 pairs. Corn has 20. Other organisms have other combinations of chromosomes. Genes are specific sets of those instructions that tell cells generally how to make proteins. You and I have about 20,000 of those genes. Different types of cells use different genes based on the cell type that they need or a specific time point in development. And the genomes of all humans differ very, very slightly. We are much, much more common than we are different. Those changes may have no impact at all. They may lead to part of normal variation, or they may have pretty serious clinical consequences and clinical symptoms. Let's pause and see if we have any more questions before I wrap us up. If I am going into the biotech field, what should I study as an undergraduate? And then a similar question, if I were a freshman in college, what courses would I take to accomplish these educational needs? Those are both great questions. Some of it depends on the specific direction that you think you might like to go in. Are you really interested in working with humans, with infectious disease, with animals, with plants? But in undergraduate, you want to take a breadth of classes. You want to make sure you get your strong biology classes in. You want to think about lots of those math classes, those advanced math or statistics. I told you computer programming is potentially a huge piece, so you might want to think about some computer programming courses. But then you also want to think about, what am I really interested in? Is that immunology? Is that virology? Is that anatomy and physiology? Because I'm interested in human health. So you want to take your freshman year and even part of your sophomore year looking at the breadth of things. What are you interested in? Equally important is the type of opportunities that you get to step into a lab. Pick courses, if you can, that involve you in undergraduate research. Talk to faculty to see if you can work in their lab, maybe even volunteer in their lab. Look for opportunities to see how what you're learning in the books actually translates into the real world. Look at summer internships. All across the country, universities and research institutes like Hudson Alpha have summer intern programs. Apply to them. It gives you another set of tools, another set of skills, and another chance to say, I really like this. This isn't so much for me. And then in your junior and senior years, look at the way that you begin to specialize. And can you accomplish what you need to do with a 4-year degree? In some cases, you might be able to do it with a 2-year degree if you're really focused on, I'd like to be a technician in this lab. Will a 4-year degree get you what you need to do? Or do you need to think about more advanced coursework as well? There are roles in the field of genomics and biotech at all different educational levels. And then there are people that say, I love science, but I really want to go into communication. You could be a science writer. You could be a videographer, and you could make beautiful videos that tell stories about science. Maybe you're really interested in the field of IT. Genomics has a place for you because of all the data that we store and all the ways that we manage data. So even if you're not set on, I want to be a scientist, but you like science, there may be a way to make those pieces tie together. Those are great questions, and I'm glad that you asked. Thank you for that. All right, that brings us to a close. So we've covered a lot of content tonight, a lot of vocabulary. There's a handout that we're also going to give you in addition to the handout from the slides that puts some of this vocabulary in a little bit more written format. So if you find yourself grappling to try to figure out how all these pieces fit together, that additional handout I think will be helpful. Next week, we're going to talk about agriculture. We're going to start with a quick section about when you talk about DNA variation, what do you really mean by that? And then we're going to jump into ag. We'll talk about plants. We'll talk about livestock. We'll talk about why chickens are so big nowadays. And it is not because of steroids. So I hope to see you back here next Tuesday night. Thank you for joining me for our first session of our virtual Biotech 201. Have a good night and take care, everyone. Bye.